Okay, so we're going to talk about how molecular phylogenetics can be used to cast light on the origins of the HIV virus. Okay, so a little bit of background. HIV was first, in 1982, large numbers of gay men in San Francisco started suffering from immunodeficiency diseases, and the cause was unknown. But fairly rapidly, by 1984, it was realized it was down to a virus, and the virus was isolated, and then they could characterize it. It turned out it was a retrovirus. So here's the virus particle here. This green protein, which is called N for envelope, that is the protein which is required for the virus to dock onto the surface of a human cell and infect the cell. So it has an RNA genome, which we'll talk about in a minute. Okay. Now, where did HIV came from? When it was being isolated, it was fairly obvious that it was pretty similar to a virus which is already known about, called the simian immunodeficiency virus, SIV, and that existed in several different subgroups, such as they're all the same species but slightly different. So CPZ, which was one found in chimpanzees, and SIV SM, which is a kind of monkey, which found in another species of monkey. Now, when for the SIV to turn into HIV, it has to infect a human being, of course, and the way it did that is presumably through people eating the meat of chimpanzees or these monkeys and come into contact with the blood. That, of course, isn't enough for it to be able to infect human beings. It must undergo some kind of change, some kind of mutation, which enables it to live in a different host. And this change of species is called zoonosis. And of course, that's how the coronavirus came out of bats into human beings. So it's a fairly well-established process. And with HIV, we realized there must have been 5 separate zoonotic events. So that 5 separate occasions, the SIV virus came out of the primate population and established itself in human beings. So there are 5 different strains of HIV there. HIV-1, which is subdivided into M, N, O, and P, and then HIV-2. So HIV-1, this accounts for the vast majority of cases of AIDS. These other 3, these are found in they're very rare, and they're only found in a few little pockets in the Cameroons chiefly. So they never really established themselves in the way that HIV-1M has become has created a global epidemic, a pandemic. This one here, HIV-2, this is even a little bit more different to the slightly more different. It didn't come from the chimpanzee virus. It came from the monkey virus. HIV-2 is only found in Sierra Leone and Ivory Coast, I believe. It's not such a severe disease as HIV-1, and it's largely located in that area of the world. Now, the one we're going to talk about today is this one here, the HIV-1M. Now, that has, since the zoonotic event occurred and it infected human beings, it has now diverged into 10 different clades. Okay, clades is an old-fashioned phylogeneticist word. Clade is, it's almost like a sort of like a breed of dog with 10 clades of dogs. So it's like subspecies, and the clades are given numbers, letters A up to K. You think that's 11, that's because I isn't used. So there are now 10 clades of HIV worldwide. They're quite divergent from each other, and just so the two important ones are B and C. So B and C, they account for I think 90% of all cases. They're by far and away the most common. B is the main type found in the USA and Europe. C is more found in the Southern African states and in India. I think B accounts for maybe 70% of cases, C 30% of those cases. Right. Now, why does HIV changes, and it's subdivided into, so it hasn't stayed constant, it's changed and it's evolved into 10 separate subspecies or clades. Why is that? Well, one of the reasons is, well, mutations always occur, but HIV is a retrovirus. So that means it's got an RNA genome, which is converted by an enzyme, which is coded for by the virus, called reverse transcriptase. Normally, of course, transcription goes from DNA into RNA. This kind of does it backwards, and it turns the RNA into DNA, which is then incorporated into the genome of the host. Now, reverse transcriptase is a very error-prone enzyme, and it makes lots of errors. And so, with the passage of time, more and more errors will accumulate. So, the more base changes you get from the original, that means greater the length of time. Right, so this was a seminal study done in 1990 then, and it was done by Korber et al. 1990, and Korber et al. If you Google that and the journal is Science, you'll find the article, and you can download the PDF for nothing, and you can see the whole article. But what they did was they got, I think it was 159 individuals infected with HIV. They isolated the virus from each one of those 159 individuals, and they sequenced the entire gene for the ENV protein. So, and then they looked at differences in the sequence. So if you got two viruses from these 159 individual people, and you found that there was hardly any changes in nucleotide sequence, then you would say that those two viruses had only diverged from each other very recently. If, on the other hand, there was a lot of differences in the base sequence, then you'd know that they diverged a long time before that. And so they, using very powerful computer to compare 159 samples, they eventually managed to make up a network. So like this, saying, so for example, that one and that one, reasonably similar base sequence, and they only diverged quite recently. The length of the line here is supposed to be proportional to the number of base changes that have occurred. Whereas if you look at these four here, these are going to be fairly similar to each other, quite different from the red ones, and they must have diverged twice, once at that point and once at that point. So they got a computer program to make up this diagram here, and that figure A is just the figure, that A is just A in the journal. But what these letters stand for, these C, J, H, these stand for the different clades of the HIV one virus. Okay, and this here, the consensus sequence, right, they are saying that this, they don't know what the consensus sequence is, but they're kind of using a computer to guess what it would have been. That was the original virus that jumped ship from the chimpanzee to the humans. Okay, however long ago that was. So in reality, the length of each of those lines should be, so the length of that line to the consensus and that line to the consensus should be exactly the same. They're not quite the same, I think partly because some of these 159 samples had been collected over a period of a couple of decades. I think that's why they're not all exactly the same. So what does this this mean then? Well, it shows you that the B and the D clades, they are relatively, they're more similar to each other than all the other ones, because they only diverge from each other at that point. You can see here we've got a group of these group of viruses here. They all must be pretty similar to each other because there's only a short line to that blue point there where they diverge. So there's only a few base changes there. And there's, you can probably see in A, there's another very, so there are 3 people in their sample. Remember, there's only 159 people, 3 people with A, and they had very similar sequences to each other, and they must have, the virus must have only diverged relatively short time. They've only accumulated a short number of base pair changes. Okay, so the consensus sequence here, there, and well, the idea was to find out how long these lines are. If so, for example, from there to there, if you can find out how long that line is, you can find out when the virus emerged from the chimp. Okay, so that data is shown in slightly, quite a different way, but it's the same data. If we have a look here, and this is done as a phylogenetic, as a phylogenetic tree, and along the, so every one of those lines, each one, there should be 159 lines, and that's 159 virus samples. This is calibrated in years along here. So starting off 1930 going up to the year 2000, but it could also be calibrated, it's proportional to the number of base changes. So the longer the line, the more base changes that have occurred. So if you just have a look at these 2 here, you see that little branch there, those 2 must have very similar sequences because they're only slightly different to each other, and they probably only, if you look at the scale, that's about 5 years. They probably diverged like 5 years previous to the samples being collected, and they're in the B clade. Whereas if you look at, well, let's just choose that one there. Okay, that's also in the B clade, but they must have diverged from each other quite a long time ago, because to get back to each other, you have to go all the way along this line. I think all the way back to there. So that, you have to go back all that length of time there. They must have diverged from each other 35 years ago. Okay, so you see how what this diagram means. And here, they've plotted it back to the original consensus sequence. And to do that, they needed the A one, because that, these ones are very different, so they must have evolved a long time ago. You can see that the original date there was somewhere between 1911 and 1945, and the computer model predicts the most likely date for that was about 1930. Likewise here, they think that the B split from the D here somewhere between 1920 and 1940, 1939 is the best guess for that, and so forth. Also, they had one sample which had been taken in 1959 from it's a frozen sample, and it's a frozen sample from a hospital in Africa, from a patient, and they found when they retrospectively tested it that it contained the HIV virus. They looked at the sequence of that and see where it fitted into their data. And I think if you read the original paper, it said it fitted in, I think, about 1957, according to their model. So they're getting a pretty good agreement between what they knew was around 19 then and when it should have been there. So the upshot of this work is that probably HIV virus jumped ship from chimpanzees to humans about 1930, whereas obviously nothing was seen until about 1980s. So what happened in the 50 years in between? Well, it must have been circulating in a local population in Africa, but it wasn't noticed in a rural African setting. Maybe it wouldn't be noticed if only small numbers of people had the virus. The symptoms from the virus are very diverse in different people. Medical care and so forth isn't that great, so that could be why that is there. And then obviously something happened in the interim. Well, probably I would say not in the 30s, 40s, or 50s, or even 60s, but maybe in the late 70s, something happened which enabled that virus to break out from the local epidemic and to create a worldwide epidemic, a pandemic.